In this video, we will discuss about mechanosensing in the Huntington disease, or what are the biological pathways that will get affected because of this mechanosensing in Huntington disease. So this Huntington disease is basically the mutations of this Huntington gene, especially in the exon 1, where this CAG triplet repeats will be increased, and when the based on the increase in the CAG triplet repeats, it will cause Huntington disease at different stages of life. For example, CAG triplets is around 30 to 35, it will cause Huntington disease at the late stage, and if the CAG triplets is more than 80, it will cause Huntington disease uh, at a very early stage. So there are two different models for this mechanosensing in the Huntington disease. We will go through each one of them one by one. So first one is about this when there is a mutation in the Huntington disease are there is a cytoplasmic aggregations of the Huntington gene occurs, and this usually supported by the upregulation of this ROC and ROA pathways. So it will support aggregations, cytoplasmic aggregations of the Huntington disease, and one of the functions of the Huntington disease is to activate this ACTD2 to interact with this F actin filament and prevent them from forming a rod-like structures. However, the mutated Huntington disease will interact with this ST2 and inhibit its functions to interact with this affecting filament. This ST2 interacts with the affecting filament and help in the methylations of this affectins, so that it will avoid this rod-like structures formations. However, in the mutated Huntington case, it will prevent those methylations, and that will lead to this affecting rod-like structure formations. And apart from this, what will happen is that these cytoplasmic aggregates will then co-localize within this nucleus, and that will lead to the transcription dysregulations, and those transcription dysregulations will also cause the formation of this coffin, and this phosphorylation of the coffin through this ROC and ROA pathways will lead to this interaction with this affecting filament, that will also lead to the rod-like structures formations of this affecting. And because of this, what will happen is that this rod-like structures formations of the affectin will reduce the cell motility. At the same time, the cells divisions or cell migrations will also be affected, because this aggregates, cytoplasmic aggregates, will affect the chromatin's stability also. And this is a brief overview that these mutations will mutated HTT will lead to this MHTT fragments, and that will cause the cytoplasmic aggregations. So this is one model, and in the other model is talks about the nuclear changes, mainly about the nuclear changes. So in this case, similarly, the transcription dysregulations occurs by the same almost the similar way that is mentioned in the model A, that is the mutated STT will form, and it forms the cytoplasmic aggregations, and also this mutated STT will also co-localized within the nucleus, and this mutated STT will help in this upregulation of this lamin B, and also change the nuclear morphology. And this increase in the lamin B plays a role towards this affecting cap formations. So increase in the lamin B formations will cause increase in the affecting rod-like structures formation. This leads to the affectins rod-like structures formations also within the nucleus. At the same time, the nucleus motility will be reduced because of the rod-like structures will formed from in the cytoplasmic part of the rod-like structures is formed in the cytoplasmic domain and in the nucleus domain. And moreover, after that, what will happen is that there's the interactions this affecting cap formations is also been affected by this Huntington proteins, and also the interaction also affected by this interaction of Huntington proteins and affectins uh, However, because of the mutated effectins formations, these interactions will be disrupted, and the abnormal cap functions will not be performed as efficiently as it will be performed when the Huntington proteins are produced in a native form. So this, in other way, will cause the cell lectins, affectins, rod-like structures formations through the nucleus and also in the cytoplasmic domain. And other part that is mentioned here is about this colocalization of these mutated Huntington's proteins will affect the chromatin structures also, and this will finally leads to this epigenetic changes or the transcription dysregulations, and that will leads to the upregulations of the proteins that are involved in this hunting, or that will promote this Huntington disease, that will cause problems in the brains. As although Huntington disease are found across the body, a major part of this Huntington disease is predominantly expressed in the brain. So the change in the transcription dysregulations will also affect the brain functions, and also the neuronal damages that are more commonly known symptoms of the Huntington disease. So these are the brief overview and two models of changes in the biochemical pathways because of the mechanosensing in the Huntington disease. If you need to understand more about this uh, topic, that is biochemical changes because of the mechanosensing in the Huntington disease, you can go through the reference that has been provided in the description. And before ending this video, I will just want to recall that in both the cases, cell motility and migrations will get disrupted because of the rod-like structures formations of this affecting. So thank you for your time.